Hi everyone. So in this video, we're going to talk about cell junctions. So a brief review from high school biology, there are going to be three main kinds of cell junctions. There are going to be your tight junctions. These are your seals. They're going to prevent movement between the cells. You're going to have your gap junctions. These are going to allow for movement between cells, things that are being transported between cells, and you're going to have your anchoring junctions, and anchoring junctions are going to hold them together. Generally, on the high school level, this is all you need to know, these three. We're going to be talking about them on the college level, so slightly higher than your AP biology level. Now, to do this, I want to draw out two cells, two model cells, and I don't know if you're going to find any specific cell in the body with all of these different kinds of junctions, but it will be useful to see them. So let's have, I don't know, let's have it look like this. Cell number 1 and cell number 2. I'm going to zoom out a tiny bit so you can see this. And let's say that this is the basal side of the cell. Basal, meaning this is where this cell is going to be anchored to some kind of surface. And over here you have your apical side of the cell. So this is going to be facing your lumen. So a good example of this would be in your small intestine, your villi. So actually, it's going to look something like this. Okay, that's probably more realistic. So I'm going to draw all three kinds of cell junctions here. So first are your tight junctions. These are going to hold the cell very closely together to one another, very closely. That's why we call it a tight junction. Okay, let me zoom in. Tight junction. That actually doesn't look that good, but you get the picture, right? It's going to hold it together, hold the cells together very tightly, and we're going to draw more diagrams of all of these different junctions later on. This is just a general picture. Next, you have your anchoring junctions. So I am going to draw them like this. These are going to cross in between the cells and literally anchor them in like this. So you're going to have these fibers that are part of the cytoskeleton inside the cell, anchoring the two cells in place. So there are actually two kinds of, oh my gosh, that is still thick from the last video. You're actually going to have two kinds of anchoring junctions. You're going to have your desmosomes. So up here, these are desmosomes. Desmosomes. So it's one word. Let me try to squeeze it in here. Desmosomes. And over here, we have the adherens junction. It's called the adherens junction. And the big difference between them, you can see that I changed the color. The color is not actually different in the cell. The difference is the kind of cytoskeletal element that this is attached to. So your desmosome is attached to your intermediate filaments. Your adherens junction is attached to your actin. We'll talk a little bit more about that in depth. But just a convention I'm going to use throughout this video for the sake of simplicity. When I have something in red, you can probably assume it's an actin fiber unless I say otherwise. And also, we have probably the easiest one to understand. We have your gap junction. This is an open space between the two cells, gap. And it's a little more complicated than this somehow, but we'll delve into that a little more deeply in a few minutes. So in general, these are the kinds of cell junctions we'll have. There are two other important kinds, two other different kinds of anchoring junctions, and these are going to be your junctions that anchor your cells down to a base. So rather than anchoring two cells to one another, it's going to anchor one layer of cells to another, so your basal side. And these are going to be your, I'm going to have one here, and this one is actin based. Actin. These are your focal adhesions. Sorry. And over here, we're going to have our hemidesmosomes. Okay, and just as you can see with your desmosomes, well, what did I do there? Oh my gosh. Just as we can see with our adherens junction versus our desmosome, the main difference between a hemidesmosome and a focal adhesion is the kind of cytoskeletal element that it's attached to. Your hemidesmosome, like your desmosome, is attached to your intermediate filament, and your focal adhesion, just like your adherens junction, is attached to an actin element. So this is a general summary of the main cell junctions. Now we're going to delve a little bit more deeply into each of these different junctions. Okay, so let's start with probably the most complicated of the cell junctions, your anchoring junctions. Okay, so again, I'm going to draw a model cell that looks like this, or two model cells rather. So this is their interface where they are going to interact. And just for the sake of simplicity, we're going to have your adherens junction and your desmosome right next to one another, just so we can compare them. So first things first, let's draw the adherens junction. Adherens junction on top. Okay, another word for this is the zonulae adherens. So you might also hear it referred to as zonulae adherens. So let's draw them out. So as I mentioned earlier, your adherens junctions are going to bind to actin. So this isn't really, what actin actually looks like. This is just for simplicity, again, so you understand it. And they're going to be linked together with proteins that I'm going to label in blue. Oh, again, still thick from the last video that I recorded. So it's going to look something like this. This is very simplified. There's a lot going on here, actually, if you really want to zoom in. But this is generally what's going to happen. These are your anchors that are going to hold your two cells together at the actin filaments. Now you might be asking what these blue proteins are. Well, there's actually a wide variety of proteins that this can be, but they all fall under the same general family called cadherins. Cadherins. And a very important thing that you should probably know about your cadherins are that they are dependent on the binding of the calcium ion. They're dependent on calcium binding. And the way that I remember this is that I didn't know this until way too late, actually, but the word cadherin actually comes from calcium adhering. So cadherin literally comes from the fact that this kind of binding is 100% dependent on calcium. Literally cadherin. There are going to be many kinds of proteins, especially in the extracellular matrix, that do rely on calcium as well. But cadherins are especially completely dependent on calcium ion binding. Some of those other proteins can actually use something like magnesium or any other divalent cation. Fun fact, actually, completely unrelated to the cell biology course that this is for, but a lot of the proteins in your body because they respond so well to divalent cations like calcium and magnesium, they'll actually sometimes respond to lead, which is also a divalent cation, and lead will not fit the function that that protein is binding for, and so that's often why lead poisoning leads to problems, because your body mistakes it for another divalent cation. Anyway, that's a tangent. I probably wasted your time with that, but fun fact that I know. And your adherence junction is going to be particularly common in epithelial tissue. So your skin is probably the best example that I can think of. So this is your adherence junction. I'm going to extend this a bit because I did not anticipate writing that much. Okay. Now, we're also going to have your desmosomes. Maybe it might be easier if I do it in a different color. Your desmosomes. Now, funny thing is that your desmosomes, also called your maculae adherens, it's another word for them, they're also going to use cadherins. However, they're going to use a very specific family of cadherins with very specific domain structure. So with very specific domains. Okay, and there's actually a lot going on here, so I'm going to try to simplify it as much as possible. Simplify it as much as I think you'll need. So you're going to have your intermediate filaments, just as you have your actin up here. So your intermediate filaments. I don't think they're actually going to look like that in this order, but the point is that they will bind to intermediate filaments. You're going to have actually very specific proteins binding to them. So let me see if I can zoom in a bit. What color can I use for these? Let me just use orange. You're going to have these strange bone-shaped proteins almost binding on both sides, called desmoplakin. I don't know if you're going to need to know that, but maybe good to know if you're interested in cell biology. So from here, all cells with desmosomes are going to have desmoplakins. From here, desmosomes can vary. There are a wide variety of proteins that they could use, but some common ones that are going to be used are, I'm just going to throw out some names. I don't know if you have to memorize them, and I don't know exactly how they bind. If you want to search that up, you can, but I don't know exactly how the binding works. I'm just going to draw them like this. In general, you're going to have your desmoplakin bound to some other kind of proteins. Some common ones are your, I don't know, placoglobin is one. Placoglobin, placofilin is another one. These are your binding proteins that are going to bind directly to your desmoplakin. And then you're going to have your cadherins, just as you had with your adherence junctions. You're going to have your very specialized, or rather differently organized cadherins as opposed to your adherence junctions. And I'm not an artist. I apologize for this horrible drawing, but that's what I can do right now. And if you want names of these cadherins, you can say the two very common ones are desmoglines and desmocollins. Remember that cadherins, I don't know what that was, but you just saw it happen. Remember that your cadherins are a whole family of proteins, so you can have a wide variety of cadherins. There's no, one size fits all cadherin. You're going to have a wide variety, which is why these are all general drawings. These aren't exactly what they're going to look like in your cell. These are just general cartoons to give you an understanding of what's happening. Now, a very important thing to note is that desmosomes are very, very, very good at holding together tissues that are subjected to a lot of mechanical stress. So a lot of pushing, a lot of pulling. So you're going to find desmosomes in tissues that are expected to be subjected to a lot of stress over time. So things like cardiac muscle, that's going to be very desmosome rich. Your heart keeps on beating, and it would be nice to have these desmosomes holding those cells together. You're also going to have them in cervical lining, the lining of the cervix, a cervical tissue, which is subjected to a lot of stress during childbirth. So those are the basics of the anchoring junctions. They're more or less going to be repeated for your focal adhesions and hemidesmosomes. Maybe that'll get its own video, because these focal adhesions and hemidesmosomes also have their own functions regarding cell movement, especially in single-celled organisms. But generally, when it comes to anchoring junctions, key things to note are they're literally going to anchor cells together. You have these chains of proteins, mostly cadherins, which are very dependent on the binding of calcium, which is why calcium is so important in your diet. And your adherens junctions are going to bind to actin, which I said in the beginning of this video will be shown in red most of the time. And your desmosomes are going to bind to intermediate filaments. Okay, so let's move on to the junction that might conceptually be the easiest to understand, the tight junction. So in elementary school, which is when I first learned about the cell junctions, or I guess early middle school, you kind of get the impression that the tight junction is kind of like two hands pushing against each other. They're waterproof. Wait, I'm making a hand motion, but I realized my camera's not on, so you can't see what I'm doing. So let me try to draw it out. You're going to have one cell and another cell, and they're just pushed against each other. And that's the impression that you get with tight junctions. Well, I'm here to tell you that this is wrong. This is not actually what tight junctions look like. They're not tightly held together in this way, even though the name is tight junctions. They are tightly held together, but they're not just pushing against each other with absurd amounts of pressure. And they're not completely sealed either. So if this is incorrect, you might be wondering, well, what do tight junctions actually look like? So I'm going to have, how do I want to draw this? Okay, I'm going to have this plasma membrane, and I'm making it wavy, and you'll see why. And I'm going to have another plasma membrane on the other side. Not like this. Okay? So think of it as kind of like two wavy sheets of paper next to each other. So these are two plasma membranes. Let me label this as plasma membrane 1, and this one is plasma membrane 2. And you can see that there's still a gap between them. However, we're going to seal them up, and I'm going to do this in green. We are going to seal them up using proteins. So these proteins are going to bind to very specific sites. You need 2 sets of proteins, one on each plasma membrane, and then they're actually going to, if you're kind of looking at this cell from the side, they're actually going to, and I'm going to change the size of this to make this easier to draw, they're actually going to line the cell membrane. Well, that might be a little too, a little too thick to show you exactly what I mean. That might be better. They're going to line the plasma membranes, and they're just going to hold the two together at this line. They're kind of like staplers. If you staple together a book, it's not like or not a book, but staple together 2 sheets of paper. They're not going to be held together at every single square millimeter of the paper. You're going to hold them together at certain points, and that's what's going to hold them together. So that's really what tight junctions look like. Um, they're not, completely pressed up against each other. There are gaps in between them, but these proteins are going to be able to hold the two plasma membranes together, and that's what's going to create a tight junction. Um, let's see, anything else that I'm missing? Tight junctions, another name for them, you might see them in your textbook or on your test. They're more formally known as zonule occludens. This is OCC LUDENS, occludens. So if, let's say we want to take a closer look at this binding site over here. Let's kind of zoom out a bit. Remember that these are being held together by proteins. Again, let's call this plasma membrane 1, plasma membrane 2. There are three very common, well, not very common, but three main kinds of proteins that you will see in your tight junction. The first is, as far as modern research shows at the time of this video, let's see, when am I making this? November 13th, 2020, 8:48 p.m. As of that time, the most important protein in the tight junctions, the backbone of the tight junctions, is called claudin. It's called claudin. See, again, that's thick. So let me make that thinner. Claudin. And obviously, I'm not going to show you exactly how claudin binds, but it's going to look something like this. This is just a very common diagram that you'll see in cell bio. You're going to have it binding to some other proteins, kind of like this. And in turn, these proteins are going to bind to actin. So actin is a double helix, so let's make it look something like that. That is not a double helix at all, but you get the picture. This claudin is going to be the main protein holding together the tight junction. You're just gonna have lines of these. If you imagine this as, this isn't 3D, but if you imagine looking into the page, you're just gonna have lines and lines and lines and lines of claudin like this holding together the two cells. Another common one is occludens. So let me write that out. Occludens. I can only presume that that's where zonule occludens comes from, but don't hold me to that. That's just my guess. I'm going to change the shape a bit. Similarly, it's going to bind to actin. Again, that's not a double helix. I don't even know why I try. And another one that's going to be important is JAM1, like this. JAM1. Once again, binding to some kind of protein, and it will bind to actin. These are from families of proteins. JAM is actually a family of proteins. So claudins, occludens, and JAMs are families of proteins. So you're going to get all kinds of proteins at the tight junction. It's just going to depend on your cells and where in the cell you are. Because these are very important, believe it or not, in signaling. These serve as fences that molecules cannot get through. And if you interfere with a tight junction, you open up areas between cells that shouldn't be open. So obviously that leaves the door open for many kinds of diseases and disorders. So tight junctions and the type of protein that you use are going to be very important. Claudins and occludens and JAMs are just big families of proteins that are very important. Okay, last but absolutely not least are going to be your gap junctions. This is, again, another very conceptually easy junction to understand. Your cells mainly have gap junctions when they want to have rapid movement between cells. So very important in signaling, sending signals between cells. But again, contrary to what you may have learned or thought in middle school, you're not just going to have these free open channels between cells. That's not what's going to happen. You're actually going to have to still be able to regulate them. So of course, proteins, the workhorses of the cell. Proteins do everything. They can do pretty much anything, given enough variations in shape. Your gap junctions are going to be made up of proteins, just like your tight junctions. But the structure is going to be a little bit different. So let's draw. Let's just keep the same basic skeleton drawing that we have. You have your cell 1 and your cell 2. I'm going to draw these in, let's do it in orange. So you're mainly going to have gap junctions made up of something called a connexin. Let me write that here, a connexin. Okay, and your connexins are going to be proteins that form the backbone of this gap junction. So I'm going to draw it here. Let's draw you. Yeah, let's do it like this. Okay, your gap junction. You can see that you have your gap, and your connexin kind of forms a nice little tunnel for them to pass through. And by the way, these are, each cell has its own. Why is that going under? Anyway, you can kind of see what I'm trying to do here. Each cell is going to have its own. They're called connexons. So each half unit is called a connexon. Each cell is going to have their own. They're going to bind very tightly. And that's going to be your gap junction. It's going to provide a tunnel for things to pass in and out. And these connexins can close. They can open and close so that things just don't pass whenever they want. There's some kind of regulation to them. That's why we have proteins. Now, it's actually going to be a little more complicated. So just like a tunnel, I'm going to draw an eye just to show you where we're looking at it. Let's look at it top down. So we're looking down into the tunnel of connexins, yes. Because they're actually a little more complicated than that. Connexins, if we look at it again, if we look at it as if we're looking down the tunnel, they're actually going to be 6 structural units. 1, 2, 3, 4, 5, 6. Okay, 6 structural units, and they're packed more tightly than this, but I just wanted to show it like this here. If you want to visualize it kind of like that, you can see what I mean, kind of, right? So again, I'm not an artist. I can't art. See, I already messed up. Regardless, let's say you have your substrate, like, I don't know, ATP. It's going to be able to go down here through the connexin. Okay. Key note is that they have 6 different structural units. And there are many different kinds of connexins, just like with your claudins and your cadherins. There are many different kinds of connexins. It's just going to depend on, what kind of substrate you want to pass through. They're very specific. So there are actually over 20 different types of connexins. So depending on what kind of cell you have or what kinds of substrates your cell wants to selectively allow to pass through, you're going to have different kinds of connexins here. And actually, if we were to, I don't know why I erased that part. If we take a closer look, okay, so if we imagine this as one connexin, it's like a tube, and let's say that this is the plasma membrane. Okay. Did I almost spell membrane wrong? Believe it or not, this is not just going to be some tube. Remember that cells, proteins rather, are going to be made up of chains of polypeptides. So actually what's going to happen here is connexin is a protein with 4 transmembrane domains. 4 transmembrane domains. I don't know exactly how deeply you're going to need to know this for your class, but I think it's good to know. So you're going to have your, again, we're going to remember our high school biology, your N terminus, which is one end of the protein, and it's going to cross through 4 times. So 1, 2, 3, 4. 4 transmembrane domains. So this black structure here, with your N terminus and C terminus, is going to cross through the plasma membrane 4 different times. That's actually what your connexin is going to look like. This is actually going to be a little longer than that, but you get the picture. So in general, these are the different kinds of cell junctions you'll encounter. Just to summarize one more time, you have your tight junctions, which, as we saw, are not two cells being pushed up against each other with high amounts of pressure necessarily. You're going to have proteins almost sewing them together like a thread. You have your 4 different kinds of anchoring junctions, your desmosomes, adherents, focal adhesion, hemidesmosome. Essentially, desmosomes bind to intermediate filaments, adherens junctions bind to actin filaments, and they are both reliant on calcium-dependent binding proteins called cadherins. And you have your gap junctions, which are tightly regulated channels lined with proteins called connexins. So I hope that this video helped. This is really basic. Again, none of these drawings are exactly going to be what they look like. They're all very general. And remember that everything I talked about here, unless I specify that it's a very specific protein, they're going to be families. So your cadherins, your desmoplackins, your claudins, these are protein families. And depending on your tissue, depending on your cell, depending on where in the cell, you might get different kinds of, different variants rather, of these proteins. Okay, so this was a brief review of cell junctions, and I hope this helped.